we are going now to have our panel discussion, and so thank you again for your impressive talks. Maybe we are going to start in the order of presentation, and Jacques, I had a first question for you regarding the genetic predisposition that could exist to this infection. We heard about this Neanderthal locus. You know, I didn't hear about that in your talk, I guess, or maybe I missed it. Does it cross with the genes that you described that could predispose to some severe form of COVID-19, or what about this locus?
So there are two loci actually that now have been published that are demonstrably coming from Neanderthal genomes, the one on chromosome 3 and the one on chromosome 12 with the OAS123 genes. So it seems that two independent introgression events from the Neanderthal into Homo sapiens have brought us something that make the carriers of those loci different in terms of innate immunity. So that's, I mean, it doesn't really change life for patients having or not having them in terms of COVID, because the effect size for individuals carrying that haplotype from Neanderthal or another one is not huge. But at the population level, it's easily detectable. So it really means that we inherited something from those ancestors that are present in between 2, 3, 4% of the European genomes. We inherited something that has an impact today on our response to infections. And it's fascinating to think that in this dance, in this continuous battle in thousands and thousands of years between humans and pathogens, we keep those scares, this scaring in our genome that can be favorable with one class of pathogen and maybe unfavorable 1,000 years later for another one. So it's a mix of paleogenomics, infectious disease genetics that is really fascinating, even if not directly usable for our fight against COVID today.
But you already know that these two loci actually they are encoding gene involved in the regulation of innate immunity. That's something yes. That is clear. Okay.
Yeah. It's innate immunity. It's a receptor for some pathogen features like OAS123 are really recognizing the viruses when they enter the organism, or cytokines or chemokines that are responsible to activate or regulate some innate immune cascades.
While the role of ABO genes does not appear to be the regulation of innate immune response, if I understood well.
It could be. It's less clear, because it's true that ABO locus is in part responsible for those molecules we have on the surface of blood cells and other cells, but it's also a region that has tons of what we call trans-EQTL. So those regions of the genome that are regulating the expression of other regions of the genome, sometimes on other chromosomes. And those regulators that are located in the same ABO locus, they are heavily enriched for the regulation of genes that are also important for innate immunity. So at the end of the day, it could all converge towards, again, some regulation of innate immunity and inflammation.
Okay. Fascinating. And what you said about the study of Tim Spector is that analysis of twins suggests that every symptoms, if I understood well, could be somehow genetically driven or controlled. So yeah, they used an indirect way of assessing shared heritability for COVID, because they didn't have the actual diagnostic. So they just sent questionnaires to those 3,000 pairs of twins they have in a UK twin study that were included here. And they compared the occurrence of symptoms during the period, the first wave of COVID-19 in the UK. And according to the answers, there was clearly more co-occurrence of a range of symptoms that are the paradigmatic symptoms for COVID-19 in monozygotic twins versus non-twins. So it was really an
indirect indication that those symptoms, meaning probably the susceptibility to COVID in the first place, was heritable to some extent. So it was hard to quantify it, obviously, but it was more than random, meaning that there is some kind of human genetic factors playing a role.
Okay, so all these genetic analyses are fascinating, but what I understood also is that today is too early to recommend any genetic analysis to somehow guide the follow-up, the monitoring of patients infected with SARS-CoV-2, right?
Yeah, there is no concrete clinical indication today. The clinical utility is far from proven, that's for sure, at the individual level. It's really more indication of which are the key pathways that are involved, and maybe for a drug repurposing, that's very important. For example, nasal interferon, for those that are defective in interferon production, that's definitely a lead. But for the individual patient prognosis or treatment, no, it's definitely not ready for prime time.
Okay, okay. So, Alberto, I was interested by the idea that you suggest not to vaccine twice the patients that have already been infected with the virus. This is, of course, a very important information. And is it true with every type of vaccine today?
Well, the evidence that is available relates to mRNA vaccines. In our own case, 150 subjects in our community immunized with the BioNTech Pfizer vaccine, and the data are pretty dramatic. In parallel, the study, again, mRNA vaccine, the study by Florian Kramer, and after those 2 studies, there were 3 more studies posted in medRxiv. So it's now 5 studies. And I am aware of a 6th study. And if you actually look carefully at the data, in our own unpublished data and the data of one of the last 3 studies and the discussion of our paper, we are concerned that a second jab in the symptomatic COVID subjects, patients who are given the first dose of the vaccine, a second jab might drive exhaustion. And there are T-cell data, small cohorts, and I happen to be aware of two pieces of data pointing in that direction.
So, and all the studies so far have been done with RNA vaccine.
RNA vaccines. We have not had access to samples.
What does it tell us about the efficacy of this vaccine? Is it a very strong stimulation of the immune system, or as compared to maybe adenovirus-mediated vaccine or so on? Is it too early to say that?
I believe it's too early. It may be I mean, this is wild speculation.
Yeah.
Just a big push, a strong push, whereas the adeno vaccines were born as single dose vaccine. The Oxford vaccine was born as single dose, and then they changed on that. So that's maybe more, but we don't know. It will be interesting to check.
Yeah, sure. So I was, of course, very much interested by your biomarker, this PTx3, this Pentraxin-3. It's somehow a surprise to find this biomarker, which was not, as far as I know, very well known before. You identify that as such a strong biomarker. You compare to some other cytokines that you can measure in the plasma, and it looks much stronger in predicting the outcome of the disease. In your RNA-seq, I introduced also the alarmins in my introduction. So did you find also these alarmins in your own data? Because they are also produced in very large quantity.
Yes, they were there. Chemokines were there. The alarmins were there. But because of our, there were data and by many people in sepsis, suggesting that PTx3 is a strong correlate of death in sepsis.
Okay.
It's a meta-analysis. And because we cloned the gene, of course, we are in love with it. So,
yeah.
So we thought we have something, and in general, we are interested in humoral innate immunity, and so that's why we focused on it.
So, but then the question, there are two questions. The first one is, how does it work, and why is it so severe to get so much PTx3? And the other question is, could we inhibit somehow the action of this protein?
We don't know whether it's just a bystander of the infection or whether it plays a role. It's a failed because it binds factor H in the complement cascade. So it may be a failed break. That could be a speculation. We don't know. I mean, we are doing a systematic analysis of interaction of fluid phase pattern recognition molecules with the virus, and we are also doing genetic analysis, by the way. And
okay.
Actually, afterwards I have a question for Jacques and Oliver.
Please go.
Yeah, I mean, it was fantastic talks. I mean, and there is in Aspergillosis there are genetics, and our molecule was one. We had a New England paper ages ago, and I think it has been largely confirmed in many studies. I wonder whether you crossed the genetics with Jacques. You see my point. Whether Aspergillosis, there is anything peculiar from a genetic point of view in Aspergillosis in COVID-19. You see my point. How is it true?
I see the point, and it was not too long ago, but times during with Corona, it all appears very long. But it was 2014, that fantastic New England Journal paper that you are an author on, and we were just discussing whether with 22% of our patients having Aspergillosis, that is just from a statistical point of view, it's a good order. I mean, it's a good number. You can analyze this because it's not super rare. However, all these patients are intubated and ventilated and ARDS at the time when we diagnose them. Means that we cannot obtain consent to do any genetic testing which is not already an established parameter. It would be fantastic to do so, but the majority of these patients will arrive at our hospital in the described condition. So it's not patients that we are caring for and we could obtain informed consent from everybody, and then those who deteriorate. It's not because it's they all are in that severe condition. Unfortunately, we're a little bit handcuffed there from a legal side.
So, just Alberto, one point to come back to PTx3 to end up with that. Is it easy to monitor in routine biology? Can you measure that?
It's an ELISA assay. So it's okay. Our tenet is that it's a low cost. Yeah. And I think I can mention that we have a retrospective analysis with 100. We have two studies. One is cooperative studies on blocking a cytokine, but we have a retrospective analysis in which we are correlating 133 patients, as I recall, correlating PTx3 levels with CT scans. And the data are promising.
But the point is when to do the measurement of PTx3, circulating PTx3, at what time? At diagnosis, or only in patients that are starting to require or developing symptoms or requiring oxygen supply, or at what moment is it the best moment to check the PTx3 level?
We are not I mean, the survival data I showed referred to measurement in the first 2 days after hospital admission and mortality at 28 days.
Okay.
We have done some monitoring, but I mean, we have some interesting results, but I would not be able to. The Bergamo data were a little bit more spread out in terms of sampling because of the situation. So it was 5-6 days after hospital admission.
Okay.
But the data, the survival data, are hospitalized patient first 2-3 days, 5 days.
Okay, Oliver.
Did I understand correctly, Alberto, that the higher the PTx activity or level, the higher the risk? Is that's the result? With aspergillosis, it's the PTx3 deficiency that was associated with aspergillosis risk.
Absolutely.
Okay, but I mean, I was confused already. But now, can you explain why that is?
I don't. I mean, I have no answer. I only have.
It's out of balance, yeah. Maybe
Yeah.
it should be in the golden middle. That's
yeah.
Yeah.
Thank you. No, I have no. I could draw a picture, but
yeah. Yes.
So Oliver, we haven't understood that it's not good at all to perform bronchoscopy in these patients, and then you have some difficulties to get access to the pathogen and to analyze the resistance from the beginning. So would you recommend to start treatment with a drug that is, well, maybe there are two questions actually. The first one is whether is it recommended to introduce these antifungal drugs without real proof of aspergillosis, maybe in some cases, and whether you should use first the liposomal amphotericin B, for example, because it's active even in resistant aspergillosis to azole. What do you recommend today?
The Aspergillus resistance, Aspergillus fumigatus resistance towards azoles is in the range of 2% in most studies.
Okay.
But if you are in the Netherlands, it's much higher. It's 10 to 15-fold higher. There are explanations for that. It has to do with agriculture, with flowers, and with bulb dipping. So you protect the flower by dipping them into liquid azoles. You dip them, and they are coated with azole, so that you don't, when you go to the garden center, as soon as they open again, you don't see aspergillus on the bulbs of the flowers because they are coated with azole. If one could imagine, if you plant them, then you have a perfect gradient, and since aspergillus is a soil is ubiquitous in soil, it all sounds like the experiment on I need resistant aspergillus, how do I select? That's it sounds like. So that is a potential explanation. What we could do is we want to treat earlier. So let's assume in most of our countries it's not an issue because it's just 2%. So as a general rule, how to approach these patients, we want to treat earlier. What we can do is collect samples that we usually discard and ignore in the diagnosis of aspergillosis. That would be a non-bronchoscopic lavage, or a tracheal aspirate. You can, because that's a closed system, so you can obtain these samples. But if there's an aspergillus colony in there, you usually ignore it. So in these patients, that paper as of December last year, we recommend don't ignore it, but reevaluate, and even with just the remote possibility of aspergillosis, you may be better off treating. And if you're in the Netherlands, you may use for that liposomal amphotericin B, I guess, and maybe even a prophylaxis study could be done. We're currently trying to obtain funding for a prophylaxis study, because with a frequency of 22%, you don't need hundreds of patients. So it's certainly much less than a corona vaccine trial.
Okay. So maybe I will have a last question for Guy. So in which conditions the scientists can get access to your fantastic infrastructures? Could you explain that?
Yeah, well, so it's open to everybody. It's a website, and there are programmatic interfaces for those inclined that way. So you go to it and you can use it. So the biomolecular data are almost exclusively open and free for reuse. There are tools for uploading the new data. So if I'm doing new sequencing or new proteomics, I can put my data in. The sensitive human data come, of course, with protection. So there is a data access process, and that is not us making the decision there. That's based on a data access committee that is typically overseeing the design of the study and setting up the ethics and the consent process. So there we have mechanisms for supporting that, but essentially we broker that conversation to a data access committee, and then when the data access committee responds, if it grants access, we can package data and make things available. So essentially it's built on very open foundations, and it's free for all to use, and there are increasing elements of documentation, and we will build some training materials as well soon. So, but there's a support function, so people can contact us if there are access problems. Okay, thanks a lot. So, thanks to everybody. I have to close this panel discussion. Thank you for your great talk, and Monica, the floor is yours.